Hello! Today we're going to tell you about Pickles13, a newly discovered GA bacteriophage.
So this is Pickles13. It is a Mycobacterium phyllorum bacteriophage, and it was discovered in Downers Grove, Illinois, in a moist soil sample taken from beside a pond.
The journey that Pickles13 went through was that it was isolated through an enrichment and then went through several rounds of further isolation and then was finally amplified to have a large amount of the phage. It also underwent PCR testing for clusters EA, EE, and EB, and it was determined to not be in any of those clusters. And it also underwent lysogeny testing, and it was identified to not have any known lysogens. And here are pictures of the plaques down at the bottom. Then we got TEM pictures from Indiana University, and a TEM is a transmission electron microscopy, which is the process of getting pictures of small organisms like bacteriophages using a microscopy technique in which a beam of electrons is transmitted through the specimen itself to form an image. And this is comparing Pickles13 to two other bacteriophages in our class, which were Belba, which was an EA phage, and Welcome, which was an ED2 phage. And as you can see, Welcome has a lot longer tail than Pickles13, and Belba has a lot elongated head than Pickles13, which can kind of show you how you can use TEM pictures to identify relationships between phages. Here are the only two other GA bacteriophages. Warren was also isolated at IWU. And as you can see, the phages all look pretty similar. They all have relatively short tails in comparison to other phages, and they all look really similar, which can show you how they're in the same cluster. It was then sent to University of Pittsburgh for genomic sequencing, where it was determined to be in cluster GA. And then we annotated it through the software Pecan, and we figured out that it consisted of 39,237 base pairs and 64 genes, 33 of which we assigned a function during our annotation to.
So next we have this pie chart that compares the different functions of the different genes. So as you can see here, this green section of the pie chart consists of genes that were determined to have no known function. And that was 32 out of the 65 genes, or 49% of the genes we annotated. There are also 9 genes that were related to the tails, and another 9 genes were involved in DNA replication. And then the light blue section here shows genes that are involved in interactions with the host cell. And another 6 genes were DNA binding proteins, and then a final 2 genes were involved in gene expression and signaling. And so, as you can see, we had no evidence in the genome for lysogeny, which may explain why the phage didn't form any lysogens. And then here is a genome comparison between Pickles3 and APA. And APA is another phage that is in cluster GA and has a lot of similarities to Pickles. Their genome length is quite similar. They both have a very similar number of genes, and they also have a very similar GC percentage. And a GC percentage is just the percentage of guanine and cytosine base pairs as opposed to adenine thymine base pairs. As you can see from the plaque images, both Pickles13 and APA have pretty defined edges, but the plaques on Pickles13 are slightly smaller than those of APA.
So this is the comparison between Pickles13 and Warren, which is another GA phage. So both Pickles13 and Warren have similar genome lengths, and they have about the same number of genes, and they have a similar GC percentage. Their plaques are not that similar. Warren's plaques are noticeably bigger, and they overlap quite a bit.
Next, we made a phylogenetic tree to show how our GA cluster fits into the broader range of phages. You can see that there's a great diversity of phages inside here. Our GA cluster of APA, Pickles13, and Warren are comparatively very close to an average phage. Here we can see that Pickles13 is more divergent than APA and Warren. It should also be noted that Pickles13 and APA were both found on Illinois Wesleyan. Pickles13 is more divergent than APA and Warren.
So we also compared the identity level between the bacteriophages in cluster GA. So as you can see, Pickles13 and APA are 93% identical, and then Pickles13 and Warren are 94% identical, and APA and Warren are 95% identical.
So the conclusions that we made from all this research and data is that, in general, by examining TEMs, GC percentages, FAM maps, phylogenetic trees, and percent identities, you can make conclusions about the evolution of phages and how closely related they are. And for our specific GA phages, we concluded from all of those different forms of data that APA and Warren are more closely related to each other than to Pickles13, but that all 3 are still more closely related to each other than other phages of other clusters. And a possible future research or next steps that we could take is a possible project to see if there's a pattern of GA phages around Illinois Wesleyan / in Illinois, because it's really interesting that 2 out of the only 3 GA phages were found and isolated at Illinois Wesleyan.
Here are the people and programs we'd like to acknowledge for helping us with this project. In particular, Dr. Bolivar and Dr. Alvey for helping us with the process of isolating and annotating our phages. And here's a list of references we used. Thank you for listening to our presentation.